back with our next class. In today's class, we are going to start a new chapter, that is respiration in plants. See, in today's class, I'm going to give you a basic idea regarding what is respiration and how is it going to be allowing the production of ATP in our body. So first thing is, what is respiration all about? See, respiration, sometimes you might think that when you're taking in oxygen and giving out carbon dioxide, that is what is respiration. But then that process is merely termed as breathing. So how is respiration going to be any different? Respiration basically is much more complex a process. In respiration, the oxygen that you're taking in, that is finally going to be used to break down the food inside your body. Now, to make it more specific, instead of calling it food, we should call it respiratory substrate. So during respiration, using oxygen, certain kind of biomolecules or organic substances would be breaking down or getting oxidized in our body. And due to this breakdown, the energy stored will be released. Okay, whatever the energy was stored in the respiratory substrate, due to the breakdown reaction, this energy will be released. Now, of course, the cell or the body would not want all that energy to go in vain, isn't it? So, the cell or the body would try to conserve as much of that energy into it. It would try to conserve the energy, right? So it will conserve all the energy in the form of ATP. Of course, the process of respiration is not going to be 100% efficient, and not all the 100% of energy or not the entire energy will be able to be conserved into ATP. A little bit of the energy will still be lost in the form of heat. Okay? Now, that is basically the process of respiration. Respiration means breakdown of the respiratory substrate to release energy, and that energy is going to be conserved in the form of ATP, while a part of it will be lost as heat. Is that clear? So just like in the last chapter you have studied about how carbon dioxide and water together would make glucose, respiration is exactly the opposite. In respiration, glucose would rather break down into carbon dioxide and water, into its constituents. So basically from here you get to know that this photosynthesis and respiration both are completely different or antagonistic processes. One is going to be constructive or building up. It is a building up reaction, means it is going to be an anabolic pathway, the photosynthesis. Whereas in case of respiration, it is completely different. It is completely the opposite. It is going to be a breakdown reaction where a substance is getting broken down into tinier pieces or into its constituents to release energy. So this is what we call as catabolic process. Alright, so one is a building up reaction, another is going to be a breakdown reaction. Alright, now regarding respiration, when we are talking about, let us talk a little bit regarding the types of respiration as well. Respiration can be of two types. First, on the basis of oxygen present or absent, we can have two types of respiration, aerobic and anaerobic. Now, aerobic respiration would mean the respiration occurring in the presence of oxygen. So when respiration would occur in the presence of oxygen, of course, the respiratory substrate will be completely oxidized. And hence a large amount of energy will be released from the respiratory substrate. Hence a large number of ATP will be formed. Okay, so aerobic respiration will completely oxidize the respiratory substrate, giving rise to a large number of ATP. All right. Whereas anaerobic respiration would be occurring in the absence of oxygen. As because oxygen is not present, so respiratory substrate will only break down partially. If it breaks down partially, all the energy stored in it, of course, won't be released. And in that case, the number of ATPs formed will only be a few. Only a few ATP would be formed. That is one of the reasons why most of the higher organisms you see around you all are aerobes. All have evolved to become or have aerobic respiration, not anaerobic. Because with a few ATP that are produced in anaerobic respiration, not higher life forms can sustain. Understood? Now, respiration again can be of another two types. Now this time the basis is respiratory substrate. Okay. So based on respiratory substrate, respiration can be floating and protoplasmic. Floating respiration means when the respiratory substrate would be carbohydrates and fats. In the cell, always the preferred form of respiratory substrate will be first, that would be oxidized, will be carbohydrate, followed by fats. So when carbohydrate and fats would be the respiratory substrate, we will call it floating respiration. Okay. Whereas when the respiratory substrates would be protein, we will call it protoplasmic respiration. Now, protoplasmic respiration is not going to be a favorable process because when the protoplasmic respiration occurs, as a byproduct, ammonia is going to be produced in the cell. You know very well ammonia is highly toxic, and hence when it accumulates in the cell, it will lead to the death of the cell. But you might be thinking that you are having protein so much of time. So is the protein intake going to be allowing protoplasmic respiration, or is it going to be harmful? No. See, protein is the last thing that starts burning in the cell. So protoplasmic respiration will only start in the cell when carbohydrates and fats have been diminished. Okay? And carbohydrates and fats will be only diminished in the body when during prolonged period of starvation. It would otherwise never start. So only when a person has or is going through a prolonged period of starvation, the person is going to suffer from protoplasmic respiration. Is that clear? Understood this? So this is what would be the types of respiration. Now, let me give you an idea regarding the phases of respiration. Just like you know that photosynthesis being a complex process, we can divide it into light reaction and dark reaction. Likewise, respiration can also be divided into three phases. First phase of respiration is called glycolysis. In glycolysis, glucose will be it will be breaking down into 2 molecules of pyruvic acid under 10-step process. It is a 10-step process. Okay, understood this? Second phase is going to be called as Krebs cycle. Krebs cycle is going to be a process that would be occurring in the mitochondrial matrix. Do remember, glycolysis occurs in the cell cytoplasm, whereas the Krebs cycle, the second phase of respiration, would be occurring in the mitochondrial matrix. And the third and the last phase is called ETC or ETS, electron transport chain or electron transport system. It would be occurring in the inner mitochondrial membrane. Now, for a very important thing that you should remember, see, out of the three phases that I just now told you about, glycolysis is going to be common to both aerobes and anaerobes. But the second and the third phase, Krebs cycle and ETC, would be only and strictly found only in aerobes. It would not be found in anaerobes. So that means for anaerobes, glycolysis is the only phase found in its respiration. Alright, understood? And that's the very reason that in anaerobes, because there is only one phase, so glucose will only break down partially. So that's why a very little amount of energy will be released from the glucose. Hence, only a few ATP will be formed. Alright? So I hope I have given you, I could properly make you understand the basic outline of the chapter. So now, let us start with the first phase of respiration, that is glycolysis. Alright. So first thing, glycolysis, the pathway was discovered by three scientists, Emden, Meyerhoff and Parnas. Hence, glycolytic pathway is also known as EMP pathway. Okay. Also, as I told you, it is going to be common to both aerobes and anaerobes. Hence, glycolysis is a oxygen independent process. Alright. Likewise, glycolysis, the site of glycolysis is the cell cytoplasm. Fine. These are certain important facts you need to remember about glycolysis. Alright. The site is cytoplasm. It is going to occur in the cytoplasm. It is oxygen independent process and it is also known as EMP pathway based on the discoverers. Alright. Now, let us finally see the mechanism of glycolysis. See, glycolysis is after all a 10-step process where glucose will first undergo a first phase called the activation phase, and then the rest of the half will be known as payoff phase, activation phase or payoff phase. Okay. Now, in the first phase, you can see here, glucose will be converted into glucose 6-phosphate. So for glucose 6-phosphate to be formed, a phosphate group has to be added to the glucose in the 6th position. This phosphate group comes from ATP. So in the first phase, in the first step itself, ATP is going to be utilized. Okay, that's what you will see, that in the activation phase, ATP will be used, whereas in the payoff phase, ATP will be produced. Okay. Now, the enzyme for this conversion is hexokinase. Alright. Now, glucose 6-phosphate would be isomerized to fructose 6-phosphate under the enzyme phosphohexose isomerase. Okay. Now, fructose 6-phosphate will be converted into fructose 1,6-bisphosphate. That means in the first position of fructose 6-phosphate, a phosphate has to be added, right? And that would be again contributed by the ATP. So in the first and the third step of glycolysis, ATP will be used. ATP here will be contributing the phosphate group. Okay. Do remember, in both the conversions of ATP to ATP, Mg2+ ion will be required. Okay. Now, in this step, fructose 6-phosphate to fructose 1,6-bisphosphate, the enzyme which is going to be acting as catalyst is PFK, phosphofructokinase. Phosphofructokinase is going to be the pacemaker of glycolysis. It will be maintaining the rate of glycolysis, and also because of that, this step will be known as rate limiting step of glycolysis. Do remember that, it's very important. Alright. Now, fructose 1,6-bisphosphate will now split up. It is a 6-carbon compound. It will split up into 2 molecules of, that is GAP, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Both of these are triose, okay, 3-carbon compounds, okay. So 6-carbon will split up to form 2 molecules of 3-carbon compound. Understood? Now, the enzyme here that will be allowing this split up is aldolase. Now finally, the dihydroxyacetone phosphate will be further isomerized back to GAP, glyceraldehyde 3-phosphate, by the enzyme triose phosphate isomerase. So up till this point, every molecule will be 1,1. But as soon as the dihydroxyacetone phosphate is converted to GAP, we will have 2 molecules of GAP. From here on, everything will be 2 molecules. Okay. So in the payoff phase, every of the compound will be 3C compound and it will be 2 molecules that we have to consider. Okay. Now see what will happen. Glyceraldehyde 3-phosphate would be further converted to 1,3-bisphosphoglyceric acid under the enzyme diphosphoglyceraldehyde dehydrogenase. Now this is a dehydrogenation step where phosphoric acid would be contributing the phosphate group to form 1,3-bisphosphoglyceric acid. Also, the hydrogen would be taken up by NAD+ to form NADH plus H+. Alright, so do remember in this step, 2 NADH plus H+ will be formed and hence 1,3-bisphosphoglyceric acid finally will be resulted. Okay. Now, 1,3-bisphosphoglyceric acid will be converted to 3-phosphoglyceric acid. Means from the first position of 1,3-bisphosphoglyceric acid, a phosphate has been released. released. This phosphate would be directly taken up by ADP to produce ATP. So as I have told you that in the payoff phase ATP will be produced. So this is the first step in which you can see ATP has been produced. And this type of ATP formation where a substrate directly releases the phosphate group and it is taken up by ADP to form ATP. This type of ATP formation is called substrate level phosphorylation. Okay. Now, 3-phosphoglyceric acid will be converted to 2-phosphoglyceric acid. This is simply a rearrangement step in which the phosphate from the third position is shifting to the second position. It is catalyzed by phosphoglyceromutase. Alright. Now, 2-phosphoglyceric acid will be converted into PEP, phosphoenol pyruvate. It is a dehydration step where a water molecule will be released and the enzyme will be enolase. Fine. And the last step is phosphoenol pyruvate will be converted to the final resultant of glycolysis, that is 2 molecules of pyruvic acid. This is also going to be a substrate level phosphorylation where PEP will contribute with its phosphate group to be taken up by ADP to directly form ATP. Do remember, in ATP to ADP or ADP to ATP, in both the types of conversions, Mg2+ is going to be required as a cofactor to the enzymes. Is that clear? So this is the 10 steps in which glucose will be getting converted into, one glucose molecule will be getting converted into 2 molecules of pyruvic acid. Okay? So this is the entire step of glycolysis. Now, in the next class, we'll be discussing about fate of the 2 pyruvic acid that are formed. Because in aerobes and anaerobes, the fate of pyruvic acid will be different. The pyruvic acid in case of anaerobic respiration will enter fermentative pathway, whereas in case of aerobes, it will enter Krebs cycle. So their fates will be different and that we'll be discussing in the next class. Thank you.